Well, I think we've kind of gone through a lot of different things today. We've missed a few things, but I think we've really have hit a lot of the highlights, especially of what's being presented at ESMO. And so I think this has been a really informative discussion. Before we end, I'd like to get final thoughts from all of our panelists, and I'll start with Dr. Gnant. Any final thoughts?
Yeah, I guess we live in a fascinating era again, and we have so many great news for our patients, and we see innovation in a constant pattern with the ability to prolong our patients' lives, to increase eventually cure rates, and to provide better quality of life. So this is fascinating. I do believe that the best breakthrough and pivotal large standard of care changing trials are done in academic collaboration with industry and with networks. And we are going to focus on biomarkers and response prediction because we have to, because otherwise even the most affluent countries will not be able to afford all these innovations for all of our patients in the next decade.
Very good. Joyce?
Well, I'm just reflecting that we're getting more and more tools to improve the overall outcome in the curative setting. We're continuing to make progress. In triple negative breast cancer, we're hearing here about survival in the Gephardt-6, albeit in a regimen that's not used standardly around the world, but nonetheless, addition of carbo with overall survival. We saw a hint in the Brightness trial. There's a big improvement in PAF-CR rate with the addition of the carboplatin 2 weekly paclitaxel followed by AC, a very large improvement in, there was although the data very early and preliminary, there was an improvement in disease-free survival that appears to potentially be developing with the carboplatin. So we'll hold that thought, and we need more data certainly on carbo, but it's encouraging. We've seen from the CREATE-X data improvement in disease-free and overall survival in those patients who do not have a PAF-CR with triple-negative breast cancer. So there's some benefits there. There are other agents coming along that look promising. Certainly the checkpoint inhibitors we have high hopes for. And then the PARP inhibitors, hopefully in the Olympia trial, in the adjuvant setting. And of course, HER2 positive, there's been dramatic improvement, and now we have neratinib to bring in there too and get an additional 4% at 5 years for patients. And then coming in with the CDK4/6 inhibitors, which will very likely benefit patients with at least aggressive biology who are still at risk for recurrence. And then I have high hopes that we will find the right population, those who have a PIK3CA mutation. So I think we're just continuing to make really important progress in patients who prevent recurrence.
Hope?
Well, it's hard to add to all of that, but and I certainly agree. I mean, I think these new therapies, I think what we've learned from the new therapies is that we can give targeted agents and not have as much toxicity as we put up with earlier with targeted agents that didn't work as well. So that's very exciting. I think we do need to identify the patients who are most likely to benefit. I think the name of the game for all of us is to move these drugs into the early stage setting as quickly as possible, the agents we think are highly effective, so that we can cure more women with breast cancer. And I think actually one of the most exciting things to me is to use the neoadjuvant setting as, we talk about it a lot as a laboratory, but as a way to decide who needs more versus who needs less, both with endocrine therapy with targeted agents and chemotherapy with targeted agents and the type of chemotherapy we use. We're really moving in that direction now, which I think is very exciting. And there are some alternatives to chemotherapy that are out there as well with some exciting data, these antibody drug conjugates. That's a whole nother area that's being explored where we have only the HER2 targeted agent, trastuzumab emtansine, but there are a number of agents coming down the pike for potentially for triple negative disease. So that's, I think, also really exciting. And then lastly, to really, I think, echo what Michael said about the fact that, we not to sound trite, but we are a village. We're not going to get any of these works done unless we can collaborate across countries between academics and the pharmaceutical industry to make the best possible trials that serve our patients in the best way.
Agreed. And finally, Michael, any comments?
I finish by quoting the St. Gallen's motto of this year, which was de-escalation, escalation. In patients who have aggressive disease, we know that escalation might be the way to go, double blockade, addition of neratinib in HER2-positive patients, addition of platinum in triple-negative breast cancer patients. CDK4/6 inhibitors are on their best way to be included in adjuvant trials. PALLAS trial, the early trials with ribociclib and also abemaciclib will go into adjuvant trials. That means not a chemotherapy escalation, because this is the type of aggressive disease, luminal cancer, who definitely doesn't need always chemotherapy, and we might go for an escalation with a targeted agent, which is CDK4/6 inhibition. And last but not least, de-escalation, and this is a dream of all of us, to use less aggressive therapy like chemotherapy, and we the Germans are not quite known to be an de-escalators, but we are learning from the rest of the world, and I'm happy to share my thoughts with my dear colleagues here and be part of this event. Thank you so much.
Great. Well, thank you very much. Well, thanks to all of you for your contributions to this discussion. And on behalf of the panel, we thank you for joining us and hope you found this peer exchange discussion to be useful and informative. Again, thank you very much for listening.